The surface of your tongue is not a gentle landscape. On a microscopic level, it's a battleground, a territory under constant assault. Each sip of hot coffee is a thermal shock. Every abrasive potato chip causes mechanical erosion. The citric acid in a lemon is a chemical attack. Within the protective fortresses called papillae are your taste buds, each containing 50 to 100 specialized cells. Exposed to this relentless turmoil, their lifespan is brutally short, averaging just 10 to 14 days. This high-stakes environment demands a radical solution, not for repair, but for constant, total replacement.
A taste bud is not a homogeneous unit. It's a society of cells. Type I cells, which are glio-like, primarily detect salty tastes. Type II cells house the G-protein-coupled receptors for sweet, umami, and bitter. Type III cells handle the sour sense. But the source of this society lies deeper. At the base of the taste bud, hugging the basal lamina, are the type IV basal cells. These are not taste cells. They are progenitor cells, a reserve of undifferentiated potential. They are the quiet engine of your palate, constantly dividing to create the next generation of sensory sentinels.
This renewal isn't chaotic. It is a precisely choreographed dance of molecular signals. When a basal cell divides, its fate is directed by ancient genetic toolkits. The Wnt-catenin signaling pathway, a crucial architect in embryonic development, is reactivated here to trigger proliferation. Subsequently, other signals like Sonic Hedgehog, yes, it's named after the video game character, and bone morphogenetic proteins guide these new cells, instructing them on whether to become a type I, II, or III cell. Your ability to distinguish chocolate from vinegar depends on these microscopic biochemical conversations happening right now.
A brand new taste cell is useless if it's deaf to the brain. Once differentiated, it must plug into the nervous system with absolute precision. Gustatory nerve fibers, which have been patiently waiting, are guided to form synapses with the appropriate new cells. This isn't random chance. The nerve endings and the new taste cells release neurotrophic factors, like brain-derived neurotrophic factor, or BDNF. These proteins act as a chemical handshake, ensuring a cell that detects sweet connects to a nerve fiber that carries the sweet signal. The system rebuilds itself without ever mixing up the wires.
If your sensory hardware is being completely replaced every 2 weeks, why doesn't your perception of flavor fluctuate wildly? Why doesn't apple taste consistently like an apple? The answer is a state of remarkable equilibrium. The rate of apoptosis, or programmed cell death, is almost perfectly matched by the rate of regeneration from basal cells. Your brain isn't experiencing a massive system overhaul. Instead, it perceives a steady, stable sensory organ because the population of functional cells remains constant. It's like replacing every soldier in an army one by one, without the army ever ceasing to be a functional unit. This intricate system of renewal is not infallible. With age, the regenerative capacity of many stem cell populations in the body begins to decline, and those on the tongue are no exception. The division rate of basal cells can slow down, and the molecular signals that guide differentiation can become less clear. The carefully balanced equilibrium shifts. Cell death begins to outpace cell replacement. The total number of taste buds can decrease, and the remaining ones may be smaller. This cellular process is the physical reality behind the common experience of food tasting blander as we get older. The signal itself is simply not as strong as it used to be.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.